Hello, my name is Anton Vila, and I'm a professor of genetics at the University of Coruña, Spain. I am here to present our effort called PFP Biomics, which is aimed towards the ecologically friendly mining of aquatic ecosystems for marine natural product, or MNPs. MNPs are compounds obtained from marine organisms that have therapeutical or other useful applications of commercial value. PFP Biomics means prospection, farming, purification by bioassays, and omics-based analysis of NMPs. PFP Biomics is part of a larger European effort called Invictus, which stands for mining novel bioactive compounds from aquatic sustainable sources. The Invictus consortium is composed of 15 European universities and research centers, plus 6 industrial partners, all involved in research aimed at discovering new applications for compounds obtained from marine sources. In keeping with the EU-promoted research philosophy as expressed in the H2020 research funding program, Invictus will uphold environmental sustainability at each step of its prospection, research and development, and marketing pipelines. Invictus's research is divided into 3 main pillars: the marine invertebrate pillar, the microalgae and cyanobacteria pillar, and the brown seaweed pillar. For all 3 pillars, an initial phase of prospection for novel chemical compounds, or MNPs, which carry potential activities of interest, is followed by a series of screens to detect such activities by means of quick bioassays. The consortium also possesses advanced encapsulation technologies, which include nanoparticles. As a result of Invictus's pipeline, a new generation of MNP-derived pharmaceutical, nutritional, cosmetic, or other products of interest will be released to market. PFP Biomics contains the marine invertebrate pillar of Invictus. PFP Biomics can function independently of the rest of Invictus's pillars, and now we're going to see what kind of things PFP Biomics can do. First, let's see what the global needs are. The world definitely needs more antibacterials, more antivirals, especially this year, and more anti-cancer drugs. Alarming news on the rapid spread of antibiotic resistance has been common in the last few years. In fact, the antimicrobial resistance crisis will probably be the next pandemic. Regarding the COVID-19 pandemic, antimicrobial resistance is also an additional burden on top of the problems created by the virus. On this front, the current medical crisis created by COVID-19 makes it all the more important to speed up the efforts to find new antivirals. Regarding cancer, the global picture is also terrifying, with a dramatic increase in cases worldwide from 19.3 million in 2020 to almost 30 million that are predicted for 2040. Low-income countries are predicted to suffer the highest increase in cases. What are the solutions? The oceans provide a plethora of solutions to the problems afflicting mankind. New compounds with antibacterial, antiviral, and anti-cancer activities have been obtained from marine sources. At PFP Biomics, our main objective is the rapid identification and chemical and biological characterization of new natural substances from marine organisms containing biological activities of interest. We call these marine natural products, or MNPs. We work together with marine conservationists that guide our prospection efforts and help us transmit the message that gaining knowledge about the arsenal of useful compounds present in natural ecosystems is key to their conservation, from ocean to pharmacy. Our Prospection Team at the University of Yucatan in Mexico identifies and collects marine organisms following the issuing of all necessary permits from the corresponding authorities. Our prospection activities are supervised by our conservation counseling team to ensure that we operate in harmony with the global need for conservation of marine ecosystems. Leading this conservation counseling team is Roberto Cerda, Director of the Oceans Division of Naturalia and founder of Restore Coral Project. Sustainability is ensured by cultivation. Our partner, Reef Water Coral Farm, can currently grow more than 40 species of tropical corals and sponges in conditions mimicking those in their natural habitats. Our bioassay-based purification pipeline is led by Dr. Carlos Jimenez from the University of La Coruña, and this pipeline is designed to quickly identify MMPs. Starting from quick extracts, we subject extracts to a series of antibacterial, antifungal, anti-cancer, and antiviral screens, whose aim is to detect the presence of bioactive MMPs. Purification of MMPs from positive extracts starts by bio-guided fractionation, which might require several rounds, after which pure MMPs are isolated. For MMPs that can be isolated in large quantities, the process can be scaled up to obtain enough material for industrial scale production. In other cases, structural characterization followed by chemical synthesis is directly performed by PFP biomics. We have shown that our pipeline leads to the quick identification of interesting MMPs. We have found MMPs with antibacterial, antiviral, and anti-cancer activities from a single prospection trial at Yucatan, Mexico. Out of 64 organisms tested, we got antibacterial activities from 8 of them. 11 showed antiviral activity and 24 a clear potential as anti-cancer compounds. As a result, we are applying for international patents. We have seen the presence of PFP biomics. We are going to look into the immediate future. As important as identifying potential therapeuticals from the ocean is understanding how they work. By using the latest omics-based technologies, we will set up pipelines to measure the activities of MMPs in regard to global gene expression in vivo preclinical studies. Out of all the omics, transcriptomics measures gene expression at the level of mRNAs or transcription, proteomics measures gene expression at the level of protein, and metabolomics does the same thing at the level of metabolites. Combining all three of them results in a complete readout of what's going on in a living system in response to the application of MMPs. Omics-based measurements of global gene expression is just like grinding meat, but instead of making sausages, we take the transcriptome, the proteome, or the metabolome, or all three of them. The transcriptome is obtained by next-generation or massive sequencing. The proteome is obtained by high-throughput mass spectrometry, and this technique, high-throughput mass spectrometry, is also used to obtain the metabolome. The omics team is composed of Anton Vila from the University of La Coruña, Dr. Mariana Leguía from the Pontificia Universidad Católica de Peru, and Dr. Susana Bravo from the University of Santiago de Compostela, Spain. Our group has the knowledge and equipment to carry out preclinical assays aimed at testing the potential of promising MMPs as therapeutical compounds. From the ocean to pharmacy, PFP biomics, biomedical solutions obtained from marine organisms. Goodbye.